how do you solve gene activation problems? When there's a gene activation problem, you start with some event in the environment, which could be heat or light or a chemical signal. Then somehow that has to result in gene activation inside a cell. There may be multiple stages of gene activation. You might repeat it with a second gene or even a third gene. Each gene activation results in some protein being produced, and then eventually that final protein causes an action or a change. I'm going to start with one big sketch, and you can pause this in order to look at the whole sketch. So you can pause this to look at the whole sketch, but then I'm going to walk you through step by step with each piece. And I'd suggest that you practice by reproducing a version of this sketch for yourself. We start with something in the environment. That could be heat, could be light, could be a chemical. And here's the chemical. If it's a chemical, it binds to a receptor protein, which is usually a surface receptor, but if the chemical can cross the membrane, it could be an intracellular receptor. That receptor, when binding happens, changes shape. When it changes shape, if it is a G protein coupled receptor, that change in shape causes the G protein to change, for example, splitting off this subunit. And that subunit of the G protein then interacts with an enzyme. That enzyme produces a second messenger that I'll just circle and call second. They're usually small molecules that are called second messengers because the first messenger was the chemical on the outside, and so this is the second messenger inside the cell. Now, what if instead the input had been heat or light? Well, heat is random molecular motion, and so you could imagine a receptor that when it's hit by more molecules, that could be water or anything else, it's going to vibrate more violently, and that also could separate off part of, say, a G protein, this hypothetical G protein. Similarly, light can cause a change by changing a bond. A photon can change a bond that can cause a change in shape of the receptor. And again, light can also cause a G protein to change and go affect an enzyme. So now we have a second messenger inside the cell. That second messenger can bind to some other protein. That other protein could be an enzyme, it could be a transcription factor, many possibilities here. That second messenger might, so there are multiple ways it can go, but the most common one we'll talk about will be in the case where it goes into the nucleus and it's binding at a particular gene that we're just going to call, for this particular example, gene transcription factor A. So if it binds to an enhancer region, that'll cause RNA polymerase to be able to bind, and we'll get messenger RNA. That messenger RNA will go outside the cell. It'll be processed by a ribosome, and we'll get transcription factor A. Keep in mind, we could instead have decided to make this either an enhancer or a repressor. So at this stage of any problem, you have an option to either repress gene transcription factor A or to enhance it to cause expression of it. Now over to transcription factor A. Transcription factor A now is a protein, can be translocated back into the nucleus, bind, in this case, to an enhancer. Again, RNA polymerase can now bind and move along. We get messenger RNA that then moves out of the cell, and now we have protein B. And protein B can now have some action. So protein B could be a receptor, in which case protein B might move into the cell, and now we have the capacity to respond to some new chemical shape. It could be a transcription factor, in which case we'd have yet another round of transcription factors regulation. It could be an enzyme that would convert some molecule X into a molecule Y. It could be a secreted protein, in which case we'd have B available to leave the cell and be part of the extracellular matrix or bind to receptors on other cells. It could be a structural protein. For example, it might be actin, in which case we might have actin filaments growing and causing a change in shape in the cell, bulging out, for example, part of the cell, or pulling the cell membrane in some location. So if I zoom back in, you can see all of these events. And what I'd recommend is you go through, think through them all, and think about how would you use them to solve problems in which some input from the environment has to affect a cell through receptors, then activate some second messenger, change gene transcription, produce new proteins, which then can have multiple different effects.